Okay, this next section of the Unit 5 lecture will be on anaerobes. Okay, so just a little bit of terminology, and some of this is going to be kind of a review. An obligate or a strict anaerobe is only able to live in an environment without oxygen. So some anaerobe species can survive some oxygen exposure, but the strict or obligate anaerobes are killed almost immediately in the presence of oxygen. And this group is seldomly associated with human infection. A facultative anaerobe is an organism that can live with or without oxygen, but it prefers and grows better with oxygen. Microaerophilic, the organism can tolerate very small amounts of oxygen, but generally speaking, they prefer less than 5% oxygen. An aerotolerant anaerobe can live with small amounts of oxygen, but not for too long. And that's just because they cannot perform their metabolic processes that are essential to survival, so they can't multiply. And then polymicrobic refers to the presence of two or more microbes present at the same time, which is common in anaerobic infections. And then exogenous anaerobes are those that are found in soil, fresh and salt water. Anaerobes that exist outside of the bodies of animals cause exogenous infections. So most of these infections are caused by ingestion or through open wounds that are contaminated with soil. Most exogenous infections are caused by gram-positive spore-forming bacilli. So an example of an exogenous infection would be gas gangrene, which is caused by Clostridium perfringens, and botulism, which is caused by Clostridium botulinum. And then endogenous anaerobes are those that are found inside the human body. So these infections occur when microbes that are a part of the normal flora are allowed to gain access to sites that are usually going to be sterile. So, for example, the bloodstream, the brain, the lungs. So these are the more common cause of anaerobic infections. Examples of these infections include colitis, periodontal infections, and antibiotic-associated diarrhea. And this, I thought this was a cool visual representation of the different types of anaerobes. So, or just different types of organisms and their environment that they prefer. So obligate aerobes, and this is supposed to represent some type of broth media, so similar to your thio. But in a broth media, your obligate aerobes, which are those that have to have oxygen to live and multiply, they're going to grow at the top of that broth where they have access to oxygen, whereas obligate anaerobes are going to be just the opposite. They will grow in the bottom of the tube where there is no oxygen present. And then facultative anaerobes, remember, they can grow with or without, but they prefer to have oxygen. Aerotolerant, and the microaerophiles, so you've got those that either need a very small amount of oxygen or can't withstand it for long periods. Okay, normal habitat of anaerobes. So there is a list, a table in your book that has a list of anaerobes that are normally present in specific sites in the body. Most infections are the result of some sort of trauma that has allowed the anaerobes to gain access where they normally would not be. So just being aware of the normal flora for specific anatomic sites can help predict which organisms are more likely to be involved in infections near those sites. Anaerobes usually produce a lot of gas and will have a foul smell to them. So both of those can be kind of indicators that you're dealing with an anaerobic organism as well. Okay, so some endogenous sources would be in the oral cavity around the teeth, and anaerobic organisms are the causative agent of halitosis, which is just bad breath. In the GI tract, so there you would have organisms that are associated with antibiotic-associated diarrhea. And in the intestines, we've talked about C. difficile before in the orifices of the genitourinary tract. So the cervical and vaginal areas contain 50% anaerobes. In the respiratory tract, 90% of bacteria in the mouth are anaerobes and can gain access to the respiratory tract. And then on the skin, so organisms that colonize the sebaceous glands, which are those glands that secrete the oily material into the hair follicle, and the hair follicles themselves. A lot of times these are common contaminants if they're recovered from skin. Okay, some predisposing factors leading to anaerobic infections. And again, these are kind of things that could be clues to if the patient could actually have an anaerobic infection. So has there been some sort of trauma to mucosal membranes? So maybe some type of bite wound, a recent surgery. Has there been an interruption of blood flow? So has there been a decrease in oxygen to the tissues that has led to some type of tissue necrosis? And always is the patient immunosuppressed. Okay, indications of an anaerobic infection. So usually these infections are going to be purulent, which means pus producing. And if you're not seeing white cells, like say you do a direct smear and you're not seeing white cells, that absence of white cells does not necessarily rule out an anaerobic infection, because some anaerobes produce toxins that will kill white cells. Is there a close proximity of the wound or the infection to mucosal surfaces? Does the infection persist despite antibiotic therapy? So anaerobes are a lot of times resistant to many of the antibiotics used for infections associated with aerobic organisms. So say the patient had an infection, they started the patient on antibiotics, but the patient's not getting better. So that could be an indication that the infection is due to an anaerobic organism, or it could just be that the patient's on an antibiotic that's not effective at killing the organism. Is there a foul odor? And that could be on at the infection site itself, or on the plate. So did you open up the anaerobic blood agar plate, and the smell, I'm telling you, sometimes that smell will knock you over. They stink. Anaerobes really stink. Presence of large quantities of gas. So you can see bubbling or hear a crackling sound if the tissue around the infection is pushed on. Presence of black colonies or fluorescence under UV light. So again, you're examining your anaerobic blood agar plate. If you see black colonies, that can be an indication that you've got a true anaerobe, or if you're using UV light to detect fluorescence. Presence of what are referred to as sulfur granules, which are not actually granules of sulfur, but they're pus filled with granules of bacteria. And then a lot of anaerobes have very distinct morphological characteristics on the gram stain. So you see an organism that's growing on the plate, and you think it might be suspicious. As an anaerobe, you do the gram stain. If you're looking at certain characteristics on the gram stain, then you might have a better feeling about whether or not that's an anaerobe or not. Okay, collection and culturing. So specimens must be collected from the actual site of the infection, not just a swab of the surface. You want to avoid long exposure to oxygen, and so you want to transport that swab anaerobically and get it to the lab as quickly as possible. If you remember from, again, I think it was Unit 2, it might have been Unit 3 when we talked about wound cultures, they do make anaerobic transport swabs, so that if you do collect a swab of an area that the doctor suspects an anaerobe from, you can like put the swab back in this holding media that keeps it in an anaerobic environment. Gram stains of anaerobic organisms should be fixed with methanol instead of heat, which helps to preserve the morphology of white cells and bacteria. Okay, if the specimen is an aspirate, air, which is like a fluid that's pulled up into a syringe, air should be expressed from the syringe. So you want to make sure that you get all that air, any excess oxygen or air out of the syringe. And then the specimen should be injected into an oxygen-free transport tube or vial. If it's tissue, if it's a tissue sample, you want to place it in an oxygen-free transport bag or vial. And usually you want that tissue ground for best results. So we have these little devices in the lab called tissue grinders. So if you get a tissue sample, you can use that tissue grinder to grind up the tissue. If blood can be from blood swabs, which are not the best choice for anaerobic culture. So if it's collected on a swab, you either want to use one of those anaerobic swabs or somehow transport it in anaerobic conditions. Unacceptable specimen types for anaerobic culture would be sputum, voided or catheterized urine, swabs collected from throats, nasopharyngeal swabs, gingival, rectal, vaginal, cervical, urethral swabs, decubitus ulcers, feet, and other exposed wounds. So essentially any source on the human body that could or is normally exposed to oxygen would not be appropriate for an anaerobic culture. Okay, so some media. The CDC anaerobic blood agar. So this is a pre-reduced media that are manufactured, packaged, shipped, and stored under anaerobic conditions. So it supports the growth of all facultative and obligate anaerobes, but it's best for your gram-positive cocci. PRPEA, which stands for pre-reduced phenylethyl alcohol, supports the growth of almost all obligate anaerobes, so your gram-positive and gram-negatives, and gram-positive facultative anaerobes. The Brucella blood agar supports the growth of almost all obligate and facultative anaerobes, but it's best for gram-negatives. Your BBE, which stands for Bacteroides bilvesculin, supports the growth of bowel-tolerant anaerobes such as Bacteroides, and some other organisms may grow on this media to a limited extent. Your KVLB, which stands for kanamycin, vancomycin, late blood agar, supports the growth of Bacteroides and Prevotella species. Also, certain facultative gram-negative rods will grow. Your anaerobic broth, so that would be your thio, but it's an anaerobic thio or chopped meat. These broths support growth of all types of bacteria, so your obligate aerobes and microaerophiles near the top, obligate anaerobes at the bottom, and facultative anaerobes throughout. So just like the picture that we viewed a couple slides ago. And then your CNA, which stands for colistin nalidixic acid blood agar. Almost all obligate anaerobes, so both your positive and negative and your gram-positive facultative anaerobes will grow. So your lab you're not going to have all of these media in your lab. And sorry, I got behind on the slide again. You won't have all of those. You're going to select, one or maybe two of those that would help in the recovery of anaerobes. And a lot of that just kind of depends on your lab's protocol and the type of specimens that they see. Okay, incubation. After proper processing and inoculation, the specimens have to be incubated anaerobically at 35 to 37 degrees Celsius, so the same temperature that our aerobic plates would be held at, but we have to make sure that they're in an anaerobic environment. Your anaerobes grow slower, so we would want to hold those plates for at least 48 hours, usually before we even open to view the plate, but especially before we report as no growth. You can use a glove box, which, let me skip to a picture of that real quick. The glove box is a cabinet that is made with the gloves built in. It's airtight. You can use the glove box for inoculating your media. So you get your specimen in the lab. It's meant for anaerobic culture. So the glove box provides a way for you to set the plates up in an anaerobic environment. You can also use these to perform susceptibility testing. So these systems are best for anaerobic processes, but they're very expensive and they're not really practical for all labs. You can use a brewer jar or some other type of jar system to provide that anaerobic environment. We've got what's also called a candle jar, which I don't think I have a picture of, but it's literally a gallon size. It looks like a pickle jar, and you can burn a candle in the jar that kind of produces that anaerobic environment, and then you leave the plates in there to incubate. The brewer jar, let me see. But if you're using that brewer jar, like right here, you can see that it's a gas generator pack. So like with the candle jar, you have to use the candle that produces that anaerobic environment. In the brewer jar, you have to use one of those kind of manufactured systems that will achieve that anaerobic environment. The disadvantage to that system, or any of these that we're talking about really, is that the plates have to be removed from the jar to be examined. So if there, especially like if there's one in particular that you're looking for, you're going to have to break the seal on that jar, go through all of those plates to find the one that you're looking for. But you know, you might be inspecting all of them, but then you have to re-incubate at that and maintain that anaerobic environment. And then this picture is actually that picture and this one. Both of those are called gas packs. They're just two different manufacturers, two different styles. Consists of a pouch that usually, it depends on the manufacturer, 1 or 2 up to 4, I think, plates will fit into the bag. There's an oxygen removal system activated, and then the plates are placed in they're in this pouch, but then you place them in the regular incubator for incubation. Sometimes the plates can be examined without removing them from the bag, which is kind of nice. So that would be on these. You know, you can kind of view through the bag to see if there's growth on the plate or not. If water accumulates and it's just condensation, you might have to remove the plates for examination and then reseal the bag. These bags can also be used for transporting specimens. So like we were saying just a minute ago, if you have to collect a swab and you need to transport it to the lab anaerobically, this might be a way to do that. Okay, identification. So aerotolerance testing. If an anaerobic organism is suspected, so say you have a wound culture that you're examining, the aerobic plates are not growing anything at all, and then you open the anaerobic plate, you've got growth on the anaerobic plate. So that would be reason to suspect that you have an anaerobic organism. So if you suspect an anaerobe, then you have to subculture that organism that is growing on the anaerobic plate to aerobic media and anaerobic media to see if it will grow. So you take that organism from the original anaerobic plate, you subculture it aerobically and anaerobically. You come back and view those plates the next day or 48 hours later. You still do not have growth on your aerobic plate, but you have growth on your anaerobic subculture. You've got an anaerobe. So at that point, you could begin testing to get an identification. The gram stain, again, a lot of anaerobes can have very distinct morphology on the gram stain. So you would want to note the shape of the cell. Are you seeing spores? Is it filamentous, etc.? So things like that. The gram stain can be misleading sometimes though, because some anaerobes don't stain the way they should with normal staining practices. So you can get some kind of weird staining results. Colony morphology and other physical characteristics. So what does it look like on the plate? So take into consideration the color or pigments produced by the organism. Does it fluoresce? Is it causing pitting in the auger? Molar tooth, breadcrumbs, and fried egg are kind of common morphologies that you would see. with anaerobes. So if you're seeing any of those, and then again, also, does it stink? Biochemical reactions, so such as your catalase, spot indole, motility, lecithinase, and lipase reactions. A lot of those are ones that would be used for identification of anaerobes. This is an example, and I think I showed you a picture of these in one of the previous units too, because these can use they've got these made for different organisms, but it's a rapid ID system. So we have like a broth that we inoculate with a suspected organism, and pour that broth that we create into these wells. Each one of those wells is testing for a different biochemical reaction. They usually have to incubate for about 4 hours. And then we come back and we read those color reactions, and that gives us the organism ID. Okay, treatment for anaerobic infections. Susceptibility testing is not routinely performed. We usually just perform a beta-lactamase test, which tells the doctor if they can treat with penicillin class of drugs or not. So the drugs of choice for anaerobic infections are clindamycin, imipenem, metronidazole, and then ampicillin sulbactam. So usually we give them that beta-lactamase result, and that kind of tells them I can use this class of drugs or I can't. So that kind of leads them in a direction. Antitoxins can be used in cases of tetanus and botulism to help neutralize the neurotoxins produced by the organism. You can drain abscesses, remove dead tissue, eliminate any obstructions, and then oxygen can be forced into necrotic tissue, which will kill anaerobes because they can't withstand that oxygen. And then surgical therapy, so surgical drainage of the abscesses and removing dead tissue, releasing trapped gas. All of this is going to help to improve circulation, so that can help treat those infections as well. Okay, now we're getting into some of the actual anaerobic organisms. So this first group that we're going to be talking about are gram-positive spore-forming anaerobic bacilli. So all spore-forming anaerobic bacilli are classified in the genus Clostridium. So as a group, they are kind of collectively known as Clostridia. So some general characteristics of the Clostridium species: they're normal flora of the lower intestinal tract, such as C. difficile. Most come from exogenous sources. They can gain access to the body through ingestion of contaminated foods, such as meats and gravies commonly, or open wounds that are contaminated by soil. So that could be with C. botulinum or C. tetani. Catalase negative. They're very large boxcar-shaped gram-positive rods, and they can be speciated by their sugar tests, so you can kind of differentiate them based on which sugars they utilize or not, and they can be grouped based on the location of their spores. So terminal spores are located at the end of the cell. Subterminal is when they're found at a location other than the end of the cell. Subterminal is also sometimes referred to as eccentric, and then central spores are located in the center of the cell. Okay, Clostridium, so some different species of clinical significance. Clostridium perfringens is the most commonly recovered species. It causes gas gangrene, which is common in diabetics due to poor circulation. It can lead to amputation if treatment is delayed. And other species can cause this, but it is the most common causative agent. It can also cause a form of food poisoning, one form that is mild and one form that is more severe, but that you don't see as often. Clinical presentation of gangrene, the muscle will become necrotic, which just allows for deeper penetration by the organism. It contains gas. It's spread through contamination of wounds. Your lab diagnosis, you've got those boxcar-shaped gram-positive rods, large, irregularly shaped colonies that will have a double zone of beta hemolysis. They will exhibit subterminal spores on the gram stain, It is reverse camp positive, dextrose positive, lactose positive, and sucrose positive. And a Nagler plate can be used to determine if Clostridium perfringens produces lecithin. So Nagler is an egg yolk based agar that contains lecithin, and a positive reaction on that plate would show what they refer to as a cloudy opalescence. Okay, here's a picture of that double zone of beta hemolysis. So you can see the colony in the very center, and then the first ring of beta hemolysis and the second ring of beta hemolysis. So it looks kind of like a bullseye. There's the gram stain. Okay, next is Clostridium botulinum. This one is considered a potential agent of bioterror because it only takes a very small amount of the botulinum toxin to produce paralysis or death. So pathogenicity, so there is a foodborne type of botulism, something called infant botulism and wound botulism. So with infant botulism, you'll see in the patient symmetrical descending weakness or paralysis. So it's sometimes referred to as floppy baby syndrome, most commonly transmitted to infants through honey, which is why they recommend that babies, I think it's up to 1 year old, do not ingest honey. Foodborne botulism, you'll see patients that show impaired mental status, paralysis. This one can be fatal, and it's spread through canned vegetables, mushrooms, fish, home cured meats. This is why they warn against, if you've ever heard like not to buy the dented cans in the grocery store, that's why. And then wound botulism, the wound can become contaminated with spores. Okay, lab diagnosis on this one, the colony morphology would be granular and translucent. On the gram stain, you would see eccentric spores. This one has a single zone of hemolysis, and it is dextrose positive, but lactose, sucrose, and salicin negative. Toxin, so this one produces a neurotoxin that acts on neuromuscular junctions of the peripheral nervous system that can cause neuroparalytic illness. So the toxin latches onto specific nerve endings and destroys them. It prevents the nerves from stimulating muscles. This is what is used in Botox injections. So prevention of this one, just adequate canning. So if you do home canning, just make sure that that process is correct, and then cooking of foods properly. There's your colony morphology and your gram stain. There's an example of a baby with floppy baby syndrome, and just a reminder to be careful if you're doing home canning. Okay, Clostridium tetani. This one causes tetanus, where the muscles lock or constrict. So clinical presentation in this, you would have patients that show continuous muscle spasms, hyperexcitability, odd facial expressions, difficulty breathing. This one is spread when spores in the environment enter the skin through puncture wounds, and it produces a neurotoxin called tetanus-spasmin. Lab diagnosis with this one, you've got slender gram-positive rods. It can swarm on the plate. It has terminal spores that make it look like a drumstick or a tennis racket. And this one is dextrose positive, but lactose and sucrose negative. And there is immunization for this one now. There's your gram stain, where you can see those terminal spores that produce that tennis racket appearance. Okay, and then the last one in this group is an organism that we have already talked about previously, Clostridioides difficile. He used to be Clostridium. His name got changed to Clostridioides a little while back. So this is the one that caused pseudomembranous colitis, which is an inflammation of the large intestine and associated with antibiotic-associated diarrhea. It's not the only cause, but it is the most common cause. So clinical presentation with this one, patient's going to have diarrhea, inflammation of the bowel. The bowel can become necrotic, where you would see white cells and fibrin forming a pseudomembrane. It spreads through nosocomial infection, where you've got overuse of antibiotics that kill normal flora and allows this organism to take over. Your lab diagnosis, you've got very large flat colonies that will fluoresce chartreuse under UV light. Chartreuse is kind of a yellow to green. You'll have thin rods. They say it has a barnyard smell. It's indole negative, and it's most often detected through a direct stool antigen test. And remember, we're looking, this is part of the normal flora, so we wouldn't recover it from stool cultures. We're looking for those toxins, toxin A, which is an enterotoxin, and toxin B, which is a cytotoxin. So really, they've got those, the EIA, the direct stool antigen test, but probably more commonly through nucleic acid amplification testing for those toxins. So again, toxin A is an enterotoxin. It's a protein that's secreted by lysed bacteria that alters cell function or damages membranes of the intestinal tract. And your toxin B is a cytolytic toxin. It's an exotoxin that can affect red cells, white cells, macrophages, and platelets. And then you can, if you're doing stool culture, CCFA auger is a selective and differential auger for recovery and presumptive identification of Clostridium difficile. It will show yellow ground glass colonies. It turns a pink auger, so it starts out pink, it turns it to yellow by utilizing the fructose, and the pH indicator in that media is neutral red. So there's that chartreuse fluorescence. It's kind of pretty. And then colonies on your CCFA auger. Okay, this next little group of anaerobes are the gram-positive non-spore forming anaerobic bacilli. So some of your general characteristics, most of these are going to be from endogenous sources that are going to be opportunists. Microscopic morphology can vary from short rods to long branching filaments. And the group can be divided into two phyla. So a phyla is a subdivision of a kingdom that share similar characteristics. So the two phyla that they are divided into are the actinobacteria and the firmicutes. So within that actinobacteria group, the first one that we're going to talk about is actinomyces. So in lecture 1 for this unit, we talked about that actinomycetes group. So don't confuse that with the organism actinomyces. So this organism is microaerophilic, so it prefers less than 5% oxygen. You can, on the gram stain, you can see short or long filamentous rods, so they can be fine, thread-like, and branching. They can be long, beaded, or clubbed appearance in the short, so you can have a little bit of variability in the morphology there. Their colony morphology on the plate is referred to as molar tooth appearance. In older colonies of Actinomyces israelii, it's a slow grower, it's fastidious. Younger colonies of the organism can appear woolly or spider-like. This one is a commensal of the teeth and tonsils. It's the causative agent of actinomycosis, which is a painful abscess in the mouth, lungs, and GI tract. Other organisms can cause this, but this one is the most common, and it can also cause something called lumpy jaw, which is caused by Actinomyces israelii in humans, or the same infection in cattle is caused by Actinomyces bovis. With this one, you can see sulfur granules. So you've got these abscesses that break open and release pus filled with granules of bacteria. Again, they don't actually consist of sulfur. They're just named that way for their appearance. Okay, Propionibacterium is normal skin flora. This one is what they refer to as anaerobic diphtheroids. So remember, we talked about diphtheroids in our aerobic gram-positive rod lecture. And again, that's just those on the gram stain, they form those what they call Chinese letter formation that can have like the shape of a V or an L. It's a common contaminant in blood cultures, but it can cause opportunistic infections such as endocarditis. You'll have a coxoid, club-shaped, or branched morphology on the gram stain, and it is catalase and indole positive. Some other actinobacteria species, Bifidobacterium, Mobiluncus, Egerthella, just know that they're in that group. Here's an actinomycosis infection. Here's that, what they refer to as the molar tooth appearance. Okay, and then the firmicutes, the only one in this group that we're going to talk about is Lactobacillus. This is very pleomorphic. It's an aerotolerant anaerobe, and remember that just means that they can tolerate small amounts of oxygen, just not for very long. You can see some varied colony morphology. Some of them can look similar to strep species, and they're catalase negative, so you would want to do a gram stain to differentiate them from strep viridans. This one is widely distributed in nature and foods. It's normal flora in the vagina, human mouth, and GI tract. This is the organism that produces the acid that controls the pH of the vagina. It's associated with bacterial vaginosis, but also serious conditions like endocarditis and bacteremia can result in immunocompromised patients. And a lot of strains of lactobacillus are resistant to vancomycin, so some treatment options might be limited for that one. That's what it looks like on the plate. Okay, gram-positive anaerobic cocci. We've got Peptococcus and Peptostreptococcus. Peptococcus is normal flora of the mouth, upper respiratory tract, and large intestine. Peptostreptococcus is normal flora in the mouth, genitourinary and GI tracts, and can cause any type of infection. You can use antibiotic discs for identification of this one, or you can just use one of those little anaerobe cartridge panels like I showed you a few slides ago. Here's what Peptococcus looks like. This is a squamous epithelial cell, and then you've got Peptococcus on the cell. Okay, this next group is gram-negative non-spore forming anaerobic cocci. A lot of subgroups with the anaerobes. So we've got Veillonella, which is normal flora in the mouth, upper respiratory tract, GI tract, and vagina. It's associated with periodontal and soft tissue infections of the mouth. Can be implicated in bite wounds, head, neck, and pulmonary infections. You'll usually see it with polymicrobic abscesses, and again, polymicrobic just means more than one bacteria. Feingoldea is the most pathogenic of the anaerobic cocci, one of the most often isolated in pure culture. And again, just mentioning a few other species here: Megasphaera, Anaerococcus, Parvomonas, and Peptinophilus. Again, just kind of know that they're in that group. Okay, moving on to our gram-negative anaerobic bacilli. So all gram-negative anaerobic bacilli are non-spore forming, often associated with peritoneal infections following some type of disruption of the GI lining. Bacteroides fragilis is the first one that we're going to talk about. It's normal flora of the mouth, upper respiratory tract, intestinal tract, and urogenital tract. This one is the most frequently recovered anaerobe altogether. So it's seen in wounds and abscesses in body sites below the diaphragm. It's associated with bacteremia. It's the most frequently recovered anaerobe in blood cultures. It's non-motile, non-hemolytic, bile tolerant, catalase positive, indole negative. On the gram stain, it can be very pale staining, pleomorphic rod with rounded ends. It can have that safety pin appearance. You want to look for growth on your BBE auger. So the auger is yellow. The media will turn brown to black in the area around the colonies. And you can use antibiotic discs or gas liquid chromatography for identification. Okay, Prevotella, normal flora of the mouth, upper respiratory tract, intestinal tract, urogenital tract. This one produces, it has pigmented fluorescence. So under fluorescent UV light, it will produce a brick red fluorescence in some species. It is saccharolytic, which it means that it is able to utilize sugars. It's gram-negative rods in short chains or pairs on the gram stain. This one causes infections of the head, neck, lower respiratory tract, and you can see a black pigment on your KVLB media. It will not grow on BBE. Colonies of this one will also fluoresce brick red under UV, and again, you can use antibiotic discs for identification. Porphyromonas, normal flora of the mouth, upper respiratory tract, intestinal tract, urogenital tract. It's largely associated with opportunistic infections. It will show pigmentation, so black or brown colonies. It will fluoresce brick red under UV light, but not all species will do that, and it will not grow on KVLB. It is asaccharidic, which means that it does not utilize sugars. And then finally, let me show you this one. So here's your Prevotella stain. Here's your Porphyromonas fluorescence. Okay, and then lastly, Fusobacterium produces long, slender, spindle-shaped gram-negative rods, which is referred to as fusiform. But not all species of Fusobacterium will present in those fusiform shapes. Most species will fluoresce chartreuse and are indole positive. Most are relatively biochemically inactive, which means that they're not going to react on those biochemical tests that we use to identify a lot of these. It's associated with brain, lung, and pelvic abscesses. Also something called Vincent's angina, which is a progressive painful infection from the mouth and throat. And this one is caused by the bacterium, Fusobacterium, and the spirochete, Borrelia vincenti. So you have to have both of those organisms together to cause this disease. Here's a picture of Fusobacterium on the gram stain. And that is the end of the second lecture on anaerobes in Unit 5.